يعطيكم العافية. السلام عليكم. محاضرة اليوم, the immunology lecture for this week is the one that discusses antibodies. What are antibodies? Antibodies are proteins, globulin proteins that react specifically with an antigen, with antigens. That any antigen that stimulated their production, it would be that antibody. So we really define each antibody with the antigen. About 20% of the proteins in the plasma are immunoglobulins, واللي هم الantibodies. There are three types in the blood according to لما نعمل electrophoresis, and then we can see the migration according to their size and charge. We see that we have these three main types: alpha globulins, beta globulins, and gamma globulins. Those are types of globulins in the blood. Gamma globulins are the antibodies. So antibodies are the gamma globulins in the blood. So one of the gamma globulins of these gamma globulins هم 5 main classes of antibodies: IgM, IgG, IgA. Immunoglobulin IG stands for immunoglobulin. IgG, IgM, IgA, IgD, and IgE. Antibodies then are subdivided into these 5 classes based on the differences of the heavy chain. So you can see that these proteins are made of 4 parts: 2 heavy chains and 2 light chains. The heavy chains and the light chain have a portion that has to do, or that can bring this randomness to specificity عشان تعمل specific part, and then it has these constant parts that are always constant among all gamma globulins. So if you change the heavy chain وتخلي light chain زي ما هي, you are basically changing from one class to another. So you keep the same specificity الموجودة على الألفا على light chain, and you just change the heavy chain. You class switch from IgM to IgG to IgA to IgD وهكذا. So what are our antibodies' specific roles? What are the main roles for them? They are the main defense that we have against toxin molecules. نتذكر لما كنا نحكي عن مثلا tetanus toxin, كنا نحكي عن diphtheria toxin, كنا نحكي عن whatever other toxin. The only way for me to neutralize toxin, a protein, is I cannot depend on phagocytosis, because phagocytosis is random. It will not chase each and every toxin by itself. That's why toxins will cause severe damage if you don't have antibodies and you have lots of phagocytes. You need to direct the phagocytes to go and eat up all these toxin materials and neutralize the toxin from working. So once the antibody binds the toxin no longer is able to do its job. عدد الtoxins عدد حكينا بروتينات. So it might not do its job anymore, and then it will be taken up by the phagocytes. So that's the main first job of antibodies. The second job is to neutralize viruses. So viruses are just a bit larger from these toxin molecules. So I want to neutralize their ability to enter cells, to engulf them. Again, I cannot depend on phagocytosis, so I need to direct the phagocytosis, and that is done by antibodies. They do the same thing for microbes. They enhance phagocytosis of microbes. نتذكر حكينا عن charge. They neutralize the charge to enable these specially encapsulated organisms to be able to help the phagocytes to engulf them. So opsonize, حكينا نتذكر كلمة opsonize, is to make ready to eat, basically. يعني يساعد على الأكل. So opsonize coats these microbes and enhance, and then makes them edible for phagocytes, or more edible for phagocytes. الرقم 4, activate complement. That will be the next lecture, المحاضرة القادمة, which is the humoral innate immunity. And then 5, the last one, is to prevent the attachment of microbes to mucosal surfaces. ذكروا هذا بإيش أي واحدة منهم. If you remember, this is IgA. We said that IgA has to coat all the mucous membranes, which prevents attachment. حكينا البكتيريا that can cause meningitis والبكتيريا that can colonize the mucous membranes has to have IgA protease to break the IgA so they can attach. So IgA is the antibody or the immunoglobulin that helps prevent the attachment of microbes to mucosal surfaces. So let's go through the main table here. So we have these 5 immunoglobulins. The percentage of each is shown. فالأغلبية وأكبر حجم عندي منه هو IgG. IgG is the main immunoglobulin. الثاني بالحجم اللي هو IgA. IgA اللي هو بالميوكس membrane. So that's the second موجود. بس هذا بيحكي لك percentage in serum. So will you find lots of IgA in the serum? No. IgA is mainly in the mucous membranes, not in the serum. So in the serum, IgG is the dominant immunoglobulin. Probably if you take all total proteins, you might find IgA to be much more than it is shown here. IgM اللي هو the first immunoglobulin that you secrete موجود على the surface of B cells, and that's the one that you make before you class switch to any other immunoglobulin. فهذا تقريبا 10% أو أقل. And then you have IgE, the one that is present on mast cells. This is much less than 0.004%. And then IgD حكينا هذا كمان موجود على some surface of B cells تقريبا 0.2%. Serum concentration. So obviously يعني this is we don't really have to go through this, بس إنه approximately milligram per deciliter. راح تشوفوا مثلا يعني this is according to the percentage of the two immunoglobulins. So بين 1000 ل 200 ال IgA وهكذا. So sedimentation coefficient. So the sedimentation coefficient is basically how well they can or how fast they sediment basically. So this tells you basically how large they are. So عادة أنا بشرح هذول التنتين مع بعض. So the sedimentation coefficient and the molecular weight really come together. So if you see, IgG is small, IgD is also small, IgE is also small. So they have very small sedimentation coefficient. They are smaller and they have a much lower sedimentation. So هذا زي مثلا حكينا مثلا كيف الريبوسوم 16S و 23S و 40S و 50S وهكذا. So this is the same thing. So IgG will be 7S, IgD will be 7S, IgE will be 8 تقريبا. IgA, we will see that IgA has 2 forms. So it has a form that is about 170 molecular weight, which follows suit with other coefficients, about 7S. And then you have another form, which is 400, about triple almost, that is 11S. This 11S form is the one that is present in the secretions. This one is the one present in the serum. So the one present in the secretion is larger, and the dimer form عادة. So معمول من تنتين يعني 2 7S's مع بعض معطوك 11S. So the structure here, all of them are monomers. All of them are monomers except 2. All of them have monomer. تمام. Now we have 2 cases where it is not a monomer. IgM is a monomer as a surface receptor for B cells. Once it is secreted, it is secreted as a pentamer. So in the serum شو راح تلاقيه؟ pentamer. هاي أولى. تاني IgA زي ما حكينا هون, we have 2 forms, 2 molecular weights, monomer, dimer. So the dimer would be the one that is present in the secretions أكثر عفوا بالmucous membranes and the secretions, yes. So this is the dimer form. Obviously the 900 and the 19S, the 900 molecular weight, the 19S would be the pentamer of IgM, because IgM is only present as a pentamer in the serum. تمام. Which of these هذا السيمبل تبعهم تمام. Which of these fixes complements? So helps complement fixate and starts the complement cascade. Which of these is ذكر من المحاضرات وحكينا IgM and IgG are the ones that fix complements. Any other do not fix complements, and that makes sense because these are the dominant ones in the serum and that are the dominant ones that we secrete against microbes. تمام. Of all these, which one that we said that goes to the placenta and covers the baby for 6 months? The IgG. Does the baby's secretion have IgA? مين يحكي لي؟ So this is نتذكر حكينا the baby's milk, the mother's milk has IgA. So the mother does provide the baby IgA. Does the IgA in which form? In the dimer form. Why? Because it's in secretions. True secretions, saliva, milk, tears. So the mom will give the dimer form, which does not go through the placenta, in the milk, which will cover the baby's mucous membranes. So the mom, as long as she is feeding the baby, مثلا احيانا قديش اكثر شيء اذا بتعرفوا قديش اكثر شيء البيبيات بدهم ياكلوا مثلا breastfeed from their mom. How long do babies breastfeed from their mom? يعني the max اكثر شيء عاده سنتين ونص بالكثير هذول سنتين ونص يعني بالكثير 3 سنين. And they are, so all those 3 years the baby will be covered by his mom's IgA in his mucous membranes عادة the gut basically. وهذول الأطفال عادة that breastfeed the longer have better outcomes, especially when it comes to مثلا autoimmune disease or asthma or inflammatory illnesses. They have better outcomes. كثير أشياء مش بس هذول diseases. So transplacental passage through the placenta أصغر واحد اللي هو the dominant one, the IgG is the one that goes through. إحنا عندنا بالسيرم IgM is pentamer فما ما بدخل. ال IgA is بالسيرم is a dimer ما بدخل. IgD and IgE هذول على الماست cells ف they are not really, they are present ما راح يدخلوا, and this is present on B cells فما راح يدخل. So obviously the only one that you will find to go through the placenta basically يا IgM يا IgG. مين منهم راح يكون IgG because the smaller one and it's not in the pentamer form, it is only in monomer form. تمام. mediation of allergic response. مين كان يشتغل على الهابتن؟ مين كان يعمل له؟ مين اللي كان إذا بيجي عليه هابتن بيعمل له cross linking أو هذا بنعرفه. This is obviously, this is something that we know. This is IgE. So IgE is the one that mediates allergic response, histamine release, present on mast cells. حكيناها. You cross link it and then you cause كل هذا حكينا. Found in secretions mean obviously IgA. This is what's in your mucous membranes, in saliva, in tears, in milk, and so on. Opsonization. مين بيعمل opsonization؟ المفروض IgG and IgM both opsonize. But IgG directly opsonizes. IgM opsonizes indirectly by activating complement, which produces C3b, which is an opsonant. So it does opsonize, but does not opsonize by itself. It fixates complement, which opsonizes the bacteria directly. So it's indirectly opsonizer. مين الانتيجين ريسبتور على البي سيلز؟ IgM. IgD حكينا في بعضهم ممكن ومش ممكن مش معروف يعني. This is still a question mark here. Polymeric form contains J chain نحكيها بعدين كمان شوية. It's basically IgA and IgM. So that's the table. هاي محاضرة خلصنا. That's all that we need to cover. So if you know everything from here on in, you are solid. يعني عرفت بالantibodies. Now we just want to cover more details here and there to cover our tracks for other purposes. So موني في عندنا كلمات اسمها هاي بالأدوية كثير بيجي تسمعوها مثلا بنتهوا الاسم الدواء ب MAB مثلا infliximab. MAB basically stands for monoclonal antibody. So using an هدول كثير أدوية اللي بنتهوا بالMAB, a lot of them are immune modulators or basically these are antibodies that are directed against a receptor to make it work or to make it or turn it off. So uh, what is monoclonal, what is polyclonal? Using an antigen in an animal to produce antibodies will yield heterogeneous antibodies. These antibodies will be formed from several different clones, cell clones. فخلينا نسأل السؤال التالي. If I give you an antigen, will your antibody be identical to an antibody that من زميلك أعطيته نفس الانتيجين, will they form an identical antibody? I'm not saying that they won't form another antibody. They won't form an antibody in a normal situation, but you will make your antibody according to which cell that you will find that can react to this antigen, and زميلك وزميلتك, whatever, all other people will do the same. So people will have at the end you will have an antibody that is formed from one B cell. انت بالنسبه لك from one B cell. تذكر حكينا كلمه clonal expansion. So once you do clonal expansion, once you have activated the B cell and you have caused it to activate, and you make all other antibodies that form this, all other cells that form this antibody all only come from this clone. فمعناته كل الانتيبوديز that you form against this antigen come from one single clone. فمعناته كلهم are identical جواك انت. So you have formed a monoclonal antibody. فأنا this is monoclonal. هلا إذا أنا باجي باخذ antigen, فبالعكس شو يعني polyclonal؟ Polyclonal يعني comes from different clones. So if you take these antibodies from several plasma cells clones, يعني أخذت اللي أنت عملتها in a specific instance. عملت لها clonal expansion, وعملت واحدة ثانية ولقيت الواحدة الثانية that can form an antibody to this, وثالثة ورابعة. So this is the first. لسه ما maybe you did not activate your B cells yet. Maybe you're still on the IgM phase. Maybe you're still making different kinds or different copies or different specificities. بس كلهم antibodies ضد هذا الانتيجين, but they are all not identical. So they come from different clones. So these are polyclonal antibodies. Both of them work. Both of them will attach to the antigen, but they don't come from the same source, basically not from the same cell. كلهم من different B cells that all react to the same antigen. So that's monoclonal, polyclonal. So if we make antibodies from one single clone, مثلا one plasma cell, for example, plasma cell tumor or myeloma, these are homogeneous, and that's called monoclonal. They are made from the same cell. So if I want to make إذا أنا مثلا بدي أعمل antibodies for research purposes or for diagnostic purposes, شو بدي أستخدم؟ polyclonal or monoclonal? I would want to use monoclonal. Why? Because I want to standardize and have the same reaction each time. إذا بستخدم polyclonal يمكن واحدة من الانتيبوديز هالpolyclonal يكون قوية، الثاني أضعف شوية. All of them work, but at different rates. So I choose the best one, the one the body chooses and makes a monoclonal form out of. هذا اللي بستخدمه أنا, and I then خلاص I use this tumor cell to this myeloma, and I produce all the antibodies that I want against this. مثلا الانتيجين بدي أعمل killing reaction, بدي أعمل دواء, بدي أعمل whatever I want to use, مثلا ELISA, whatever I'm trying to do, I will use the same monoclonal antibody. مثلا دواء حكينا نفس الشيء. So monoclonal antibodies also can be made in laboratory. How do we make it? We bring a myeloma cell. This is طبعا الخلية هاي بتكون immortalized. It's a myeloma cell, obviously, that is able to produce antibodies, and then this myeloma cell, and then we fuse it with an antibody producing cell. We fuse them together, and this is called a hybridoma, and then we make lots of cells of this hybridoma, which all are the same clones. And then we make a large quantity of antibodies, and then هذا هو نستخدمه for diagnosis, for research. دائما الحين مثلا نجيب antibody لهاي الشغله وantibody لهاي الشغله. Basically, I just bring an antibody producing cell against this antigen that I want. I fuse it with a myeloma cell, and then I make خلاص بصير عندي endless supply of these cells, and then I keep growing them and collecting the protein that they make, which is basically the antibody monoclonal antibody of my fusing. Structure. Antibody structure حكينا 2 light chains, 2 heavy chains with all constant parts except the top parts are variable. So I have a variable heavy chain, another variable heavy chain, a variable light chain, another variable light chain. This is where all the variability is done. So I can have different variability to different antigens, and everything else is a constant part. So I have in a heavy chain 1 constant part, 2 constant parts, CH1, CH2, and CH3. Same on the other heavy chain, and I have only 1 constant part. Sorry, where was I? Okay, and then 1 constant region in the light chain. So, so here we have disulfide bonds between CH1 and Cl, and then 2 disulfide bonds between CH2 and CH2. Which of these activates complement? CH2 on the heavy chain is the one that activates complements. هاي الفراجمنت اللي هي معمولة من CH1 و CH2 is called the FC fragment. تذكروا حكينا كلمة FC fragment. This is the FC fragment where the هلا الانتيبادي من هون من هاي الجهة بمسك بالانتيجين من هون، من هاي الجهة هذا شغل مين؟ هذا شغل الفاجوسايتس. الفاجوسايتس ما لهم بهاي الجهة، ما بيعرفوا الفاريبل region because it's always different، شكله مختلف. بس they know this region because this region is the region that is always the same. So they know this region, they interact with this region. This is the region that binds cell surface receptors. It's one اللي هم مثلا بالفاجوسايتس, neutrophils, macrophages, whatever. So all the cells of the body that wants to deal with this antibody, they deal with this FC portion. هاي جهة الإدارة تبعتنا. This is the antigen binding area اللي هي FAB fragment أو FAB. So اللي هي معمولة من variable region. شو ضل هيك وانا اشياء نحكيها. So this is the amino acid terminal end, or this is the carboxy end. So okay. تمام. Immunoglobulin structure حكينا. So immunoglobulins are glycoprotein made up light chain, heavy chain, تمام, Y-shaped, identical H chains and 2 identical L chains. So I just want to, if I this chain and this chain is identical. High chain وهاي chain is identical. So it's basically it's made twice, يعني, and then it's bound together. Identical refers to the gene. If each product is identical, the diagnosis comes from the same gene. تمام. Antigen binding site. وين راح تكون antigen binding site? Obviously in the FAB portion. So this is the hypervariable region. This formed about from 5 to 10 amino acids. هيهم هدول hypervariable regions. This is all a variable region. So very hypervariable. That makes the antibody different each time. So you can have different variability to different antigens. So heavy chain hypervariable regions are present all over the variable region on the heavy and the light chain. So 3 of those are even كمان extremely variable, and most of the specificity is due to these amino acid differences in here. The antigen binding site basically will bind to these hypervariable regions of the light chain and the heavy chain. والانتيجين راح يكون بيناتهم من هنا. According to these amino acids at these different regions, this is what determines the specificity of this antibody, this variable region to the antigen. So how are hybridomas made? I think we went through it already. Animal is immunized. Okay, so these are the steps. So first we immunize the animal with the antigen. نحط الانتيجين بالanimal. We take spleen cells from this animal and then we culture them with حكينا معهم myeloma cells, and then myeloma cells are able to grow independently, but they don't produce any antibodies. But we put them with these spleen cells, with the fusion happen, and then we promote this fusion by adding certain growth factors and chemicals. And then these cells are grown in special culture medium that selects only for the fused cells, and the parent cell will die, so only fused cells will remain. And these fused cells then start making screen for the production of the antibody for the antigen of interest for me. So spleen cells from the animal that is immunized plus myeloma cells نحطوا مع بعض. They are fused. Fused cells are selected in special medium that unfused cells will die, so only fused cells will remain, and then a single hybridoma cell is placed in a well and monitored for production of monoclonal antibody against this antigen. بعد ما ادور بيناتهم ودور اي وحده منهم كانت the one that I want. So chimeric monoclonal antibodies, chimeric proteins are mainly proteins made out of two or more fragments that are sourced from two or different more species. فمثلا باخذ fragment من human fragment من mouse، هذا بصير اسمه chimeric protein. هذا هو الكاميرا اسم بالقصص القديمة الإغريقية، إنه إذا بتعرفوهم هذول الوحوش القديم بيحكوا عنها، مش عايش أكيد يعني، بس إنه وحش نصه أسد ونصه إيجل سموه، ولا شيء ثاني اللي هو كان والله ناسي زمان قرأتهم. I don't recall, something like a bull and a human or something like that. So different species, 2 different species, 3 different species. So these 2 هذول كانوا يسموها كاميرا، فمن هاي الكلمة طلعت كاميرا اللي هم أنا طالع لي البروتين جاي من 2 different species, mouse and human basically. أغلب الأوقات mouse and human. That's what we use. So the basic idea is that the constant region is from a human, and we tailor the variable region in an animal. So we get the human FC portion ونجيب ال FAB portion من mouse. فأنا عندي ال FC portion عشان أنا خلاياي ما بتعرفوا إلا على ال FC portion، وإذا بدهم يعمل antibody ضدهم يصير عندي أنا antibodies against foreign antibodies راح يكون إذا كان mouse، فإذا بحط أنا ال FC portion تبعت human ما راح أعمل antibodies ضدهم. So using human constant chain will allow complement to be activated لأنه يكون CH2 موجودة. Also we prevent antibodies being formed against this monoclonal antibody. So, and then I can use a variable chain from a mouse that is much easier to obtain. And then chimeric antibodies can kill tumor cells مثلا، can بيعملوا مثلا by complement mediated cytotoxicity because they can allow complement to activate. They can or deliver toxins مثلا specifically to tumor cells. مثلا ال FAB portion بتمسك على ال tumor cells and I can bind it to a toxin مثلا. وهكذا. This is chimeric monoclonal antibodies. So important functions of these immunoglobulins. I think at this point we know all of these. So let's just go through them. IgG is the main antibody. Okay. Opsonizes bacteria, making them تمام. Fixes complements نعرفها الحكي من التable. Neutralizes bacterial toxins نعرفهم. Viruses نعرفهم. Crosses the placenta نعرفها تمام. IgA is the secretory one. Prevents attachment تمام. Does not fix complements نعرفها. IgM produces primary response. هذا أول شيء بيطلع من البي cells. It's a receptor. Before we change the chains and we switch to IgG. So it does not cross placenta. Antigen receptor on the surface of B cell عارفينها. IgD لسه uncertain, found on surface of many B cells. We know this. IgE immediate hypersensitivity type 1. Basophils and mast cells defend against worm infections. تذكروا حكينا they bind these cells so they can eosinophils will come and release their enzyme. We know this. Does not fix complement. We know it. Main host defense against filament infections. Okay, so we know everything in here. Now this table is in our repertoire. تمام. The classes IgG. Each IgG consists of 2 light chains and heavy chains linked by disulfide bond. تمام. Because it has 2 identical antigen binding site, it is called divalent. تمام. There are 4 subclasses. The IgG نفسه it's subclass into 3. IgG 1, 2, and 3, and 4, based on antigenic differences in the H chains, so in the heavy chains. So, and then the number and location disulfide, أغلبهم, the majority of IgG is IgG1. IgG2 is usually done against polysaccharide antigens, and this is important against encapsulated bacteria. IgG also crosses the placenta, not but only to the side, because of the FC portion of the placenta as well is directed to placental cells. بيمسك بالplacental cells, الplacental cells بيحملوه بدخلوه جوا البيبي. So it is therefore the most abundant immunoglobulin in newborn. IgG is 1 or 2 immunoglobulins that can activate complement. Again, you know this. تمام. IgG can opsonize enhanced phagocytosis because there aren't receptors for it. The heavy chain تبعها on the surface of phagocytes. So the receptors are there. Phagocytes, they only know the FC portion تبعت IgG. فهم بدوروا على FC portion تبعت IgG ما بيعرفوا الثاني. عشان هيك they can only opsonize the only one that opsonizes is IgG. IgG حكينا indirectly does it by ايش؟ It activates complement and then can make من complement activation بصير في عندنا C3b. C3b opsonizes bacteria. So this is the indirect pathway. Subclasses 1 to 3 are more effective opsonizers than 1 and 3 أحسن من 2 and 4. تمام. On IgG heavy chain, different sugars are attached. So if the IgG molecule has a terminal in acetylglucosamine, it elicits the pro-inflammatory response. تمام. And this allows it to bind to MBL and activate complement. So IgG that has sialic acid will not bind to MBL, and then this will make it anti-inflammatory. Opsonins literally mean حكينا preparing something to be eaten, so they are present in the body secretions. They coat foreign objects, but innate phagocytes بيجي يمسكوها بياكلوها. So opsonins are antigens واللي هم IgG, and then IgM, and then complement. These are the opsonins that we have. So antibodies and complement antibodies هم IgG and IgM. So these are the part of the antimicrobial proteins that we have. There are other more antimicrobial proteins than this. IgA is the main one in the secretions, prevents attachment, allows antibody dependent cellular cytotoxicity by depending on the cells that are present. And then this is the serum IgA. Secretory IgA prevents attachment. So we have serum IgA, which helps in the ADCC present on by اللي هم حسب الخلية الموجودة يا mast cell يا eosinophil or by phagocyte as well. IgM is the one that you produce in antibody عفوا in B cell خاصة بال B cell independent response عفوا T cell dependent B cell response. So this is where you make IgM the most, and this is the one that you make at the beginning. You start making IgM, بعدين you class switch بتغير الجينات and start making IgG instead. بتغير السويتش تزيح التشينز بتحط تشينز لهم جداد وهم هذول بصيروا من IgM بصيروا IgG, and then it becomes forever IgG. So the serum عفوا it's a pentamer it's a monomer on the surface of B cells, and then once it is secreted, it basically secretes what its receptor. That's what it does to the B cell. The receptor has 5 binding sites, and is the most efficient immunoglobulin agglutination and complement fixation. IgD ما بنعرف عنه ولا شيء, but we know it's present on some B cells. IgE حكينا this is for immediate type 1 hypersensitivity by binding IgE on mast cells, and participates حكينا for eosinophils against parasites. Immunoglobulin genes, هلا نحكي عن الجينات, the genetic rearrangement. So the story here is very similar to the story that we mentioned in the T cell receptor. It is a bit more larger and more complex. That's it. So using a relatively small number of genes, نقدر نطلع a huge number of different antibodies. زي موجود كان بالتي cell receptor, I have 50 genes of type 1 مثلا heavy chain CH1, I have 50 genes for CH2, I have 50 وهكذا. I pick a gene from here and here and here and here, and I can make up a different antibody for each B cell as it is matures. مش حكينا as the B cell matures it is selecting its B cell receptor صح؟ It's selecting its B cell receptor and the B cell once the B cell receptor is mature معناته الخلية B cell صارت mature and the B cell is the one that becomes into a plasma cell which releases antibodies. So basically this maturation into making the B cell receptor is basically the B cell is now selecting which gene it wants. That's why it's maturing. Why it's maturing? أخذت جين من هون جين من هون أخذتهم تركبهم مع بعض. While it's doing this, it's making the receptor is maturing. Once it has made up its mind, it خلاص انتهينا. أنا هذا اللي أخذته. أنا أخذت جين 37 من هون وجين 19 من هون وجين 70 من هون وجين 6 من هون حطيتهم مع بعض. خلاص أنا are you done? Are you sure? Yes. سكر خلاص انتهينا. Now this is your B cell receptor, and that's now you are mature. So that's basically what happens in this. So mutation in these genes arrest the development of lymphocyte and result in severe combined deficiency. Obviously, if you don't have these switch genes, these are the genes called RAG1 and RAG2 recombination activating genes. This is the genes that do this DNA rearrangement. هم دول بستخدم أنا RAG1 و RAG2 in selecting. أنا selecting مثلا جين 37 جين 70 جين whatever. I use RAG1 and RAG2 to make this selection. هذول اللي بستخدمهم. If I don't have these two and then these two genes, RAG1 and RAG2, I can't make different antibodies. This will cause severe combined immune deficiency. تمام. شو هم ال regions? So I have variable regions and constant region. There is a separate gene pool for each of these immunoglobulin chains. So for the kappa light chain, for the lambda light chain, and the 5 heavy chains. Each pool contains a set of different V gene segments. شو هم البولز اللي بستخدمهم؟ V gene segment and D diversity. This is only in H chains. J joining and C constant. I have these 4 pools that I choose from. فحكينا during B cell differentiation, the first translocation IgM as the first antibody produced is the primary response. After genetic rearrangement, the rest of the gene pool that was not used is discarded. This is extremely important. And this is why particular B cell becomes committed to that one specific antibody. زي ايش يعني شو اللي بنحكيه هون احنا قاعدين؟ مش حكينا as the B cell is maturing, it chose a D gene, then it chose a V gene, and it chose an H gene, and it chose a J gene, and a C gene. تمام؟ أنا أخذت 37 و17 من هون و5 من هون و37 من هون ورقم 5 من هون بكب الباقي كله. So it can never go back and change its mind. إنه أنا لا بدي أغير السبيسيفيسيتي تبعتي بدي أعمله وكذا. It has discarded all other genes during its maturation. So this B cell becomes committed towards this specificity. So clonal expansion of that one B cell. فأنا إذا هاي البي سيل مفيدة لإلي أنا، هاي البي سيل طلعت الريسبتور تبعها actually binds. خلينا نحكي مثلا السبايك بروتين تبع الكورونا فيروس or binds the capsule تبع pneumococcus or binds this toxin مثلا. So this is a very important B cell to me. So I do the clonal proliferation for it if I activate it. Because this one is important to me. This is something that saves my life. I need more of it, more and more of it. So that's why we make a lot of that one. So prior to the release of antibody, the sugar portion is added to the H chain. حكينا تبع activation or pro-activation, pro or anti-inflammatory. تمام. So this is what is going on. V genes من VH1 لا whatever. I have lots of D genes, lots of J genes, and then constant gene. Constant gene دائما هو بيعمل constant part فما بتغير هذا. فit picks one from these. أخذت مثلا VH2 من هنا and then chose مثلا من ال D أخذت D مثلا one of these D's أخذت ال J مثلا تبعها constant, and then this is DNA rearrangement. This is the gene I made. Then the transcription RNA splicing, and then you make your IgM gene and missing IgJ, which is translated into the protein IgM. So immunoglobulin class switching, isotype switching. انت بالبداية الخلية هي البي سيل قاعدة تعمل IgM دائما تعمل IgM. So to induce a more specific response I make a second gene rearrangement which produces antibodies to different class but the same specificity. أنا ما بغير specificity لأنه أنا كبي سيل ما عندي غير only one specificity. I can't change my mind because I already committed. But I can change the هلا بورجيكم I can change the chains that I use, and then I can come up with a different class of antibody that is still the same specificity, the same of the mother IgM. So antigenic specificity remains the same for a lifetime of that B cell and plasma cell because it has been determined by these genes, no matter which heavy chain constant region is being utilized. So أنا بغير الهيفي chain constant region only. So I only change the constant region, but بتحول ال IgM هذا بصير IgG, which is something I still have. I did not discard. I only discarded the other ones اللي كنا نحكي عنهم مثلا ال J وال V وال D. So I have 5 constant parts of the heavy chain. I use the first one IgM. If I want to class switch to IgG, I stop making هذا بمسكه بكبه. That means الخلية can no longer come back to make IgM. Once it class switch out of IgM وخلاص حولت ل IgG, it can never go back making IgM. It is committed to making IgG from that point forward. Let's look at it again. So we have the variable, the direct, the joining, these different constant parts. And then many constant parts, you begin with the M, and then you have the others. The first DNA rearrangement is to make, classes. This is the one that makes classes. So you have already chosen which V, D, and J that you made, and then you have all the constant part بالبداية. So this is now, if you transcribe this, you will make IgM. فأول واحد اللي هو كان هنا هو M, mu. If you want to transcribe this in making, you put it with the constant of mu, and then you're making IgM. هلا بدك تعمل switch, second switch, تمسك هذا بتكبه بتنقي واحد من تحتيه. بدك تنقي واحد من الاي جي جيز هيهم 1 2 3 4 بدك تنقي واحد منهم. اخذت واحد مثلا you chose or A بالنهايه مثلا you chose that one. تمسك هذول من هون لحديها بتمسك هذا كله بتكبه. So now you take the variable part اللي انت you already made it during maturation of the B cell in the bone marrow مع الconstant region هي هلا استخدمت A now makes IgA مثلا. So this is an IgA producing B cell. Another one, هاي نفس الفكرة. So this is the same thing. So gene, heavy chain, locus, and IgM expressing B cell. The same thing. This is the variable part. These are all the constant part. بمسك بالنص مثلا بتتحول ل IgG1 producing. أنا كنت IgM producer. بالبداية ليش؟ أنا ليش إحنا بالبداية دائما نعمل IgM? Because this is the first heavy chain, constant chain بالgenes. So always this is the default. دائما تعمل IgM default. بدك switch, you have to activate this B cell, and then it will choose which one it wants to use. بدك تعمل استخدمت مثلا تعمل IgG بتمسك بيناتهم IgG3 and this one بتكبهم. هلا هاي الخلية تعمل IgG1 تقدر تعمل class switching ل IgA واحد. هذا السؤال الأول. تقدر تعمل class switching ل IgM. هاي السؤال الثاني. السؤال الأول yes you can class switch to anything else downstream from هنا because هذول الجينات ما كبيتهم. But I can never go back to the ones that I already discarded. مثلا ما بقدر أعمل IgG3 من IgG1. ما بقدر أعمل IgM من IgG1 because these are discarded. And then I NHJ non-homologous end joining, and then I sit on هذول excised and thrown away. تمام. Note that once the B cell has class switch, it can no longer make that class of heavy chain again. Class switching occurs only with the heavy chains. The light chains do not undergo this switching. Switch recombinase is the enzyme that does this. Concentration of various interleukins. 4 نعرفه, 5 نعرفه. So it depends on the factors to class switch. مثلا 4 for IgE, 5 for IgA. Even though a cell has 2 alleles of genes of these antibodies, only one allele is used. واحد ممكن يسأل طب أنا دكتور أنا بستخدم الجينات تقول كبيتهم، ما انا I have two alleles، ليش ما ارجع للاليل الثانيه مثلا او اغير رايي، بدي اعمل another specificity للvariable region، ولا بدي اعمل different constant region واعمل different because the other one is always excluded. I can't open it. ما بستخدم الاليل الثاني نهائيا. لهنا بنخلص محاضرة antibodies. I hope it was it's one of the more enjoyable lectures. I hope that you have captured all the objectives behind المحاضرة. Please don't hesitate to ask me anything. يعطيكم العافية.